Scientists studied the gut bacteria of people over 100 years old, centenarians in Japan, Sardinia, and the United States, and found something remarkable. These individuals did not share a specific diet, a specific lifestyle, or specific genetics, but they did share one thing: an unusually diverse and resilient gut microbiome. The research now strongly suggests that the trillions of bacteria living in your gut are not just helping you digest food, they are actively regulating your immune system, your inflammation levels, your brain chemistry, and, through several recently discovered mechanisms, the speed at which you age. This video covers the science of the gut longevity connection, what the research shows, which bacterial species matter most, and what you can actually do about it. The human gut microbiome is a community of approximately 38 trillion microorganisms, bacteria, fungi, viruses, and other microbes living primarily in your large intestine. The number of microbial cells in your body is roughly equal to the number of human cells. You are, in a very real sense, as much microbe as you are human. These microorganisms perform functions your own cells cannot. They synthesize certain vitamins, including B12, K2, and several B vitamins. They break down dietary fiber into short-chain fatty acids that nourish your gut lining. They train and regulate your immune system from birth. They produce neurotransmitters, including serotonin. Around 90% of your body's serotonin is produced in the gut. The composition of your microbiome, which species are present and in what ratios, is unique to you, like a biological fingerprint. And this composition changes throughout life in response to diet, stress, medications, sleep, exercise, and age. There are now 4 well-characterized mechanisms by which the gut microbiome directly influences biological aging. Let me walk through each one. Mechanism 1. Intestinal permeability and systemic inflammation. Your gut lining is a single cell layer thick, an extraordinarily thin barrier by producing short-chain fatty acids, particularly butyrate, which nourishes gut lining cells. When the microbiome is disrupted through poor diet, antibiotics, or chronic stress, this barrier can become permeable. Bacterial fragments, including lipopolysaccharide from gram-negative bacteria, leak into the bloodstream. Your immune system mounts a response. This produces chronic, low-grade systemic inflammation, sometimes called inflammaging, which drives cardiovascular disease, neurodegeneration, metabolic dysfunction, and accelerated cellular aging. Mechanism 2. Short-chain fatty acids and energy metabolism. When beneficial bacteria ferment dietary fiber, they produce short-chain fatty acids, butyrate, propionate, and acetate. These are not just fuel. Butyrate specifically inhibits histone deacetylase, an enzyme that controls gene expression. It modulates the expression of genes involved in inflammation, cell growth, and longevity pathways. Propionate signals satiety in the brain and regulates glucose metabolism. These molecules are, in effect, epigenetic regulators produced by bacteria living in your gut. Mechanism 3. The gut-brain axis and neuroinflammation. Your gut and brain are connected by the vagus nerve, a direct communication highway carrying signals in both directions. Gut bacteria produce neurotransmitters and their precursors, GABA, serotonin, dopamine precursors that influence mood, cognition, and stress response. More relevant to aging, gut-derived inflammation can cross into the central nervous system, contributing to neuroinflammation, which is increasingly implicated in Alzheimer's disease, Parkinson's disease, and cognitive decline. Mechanism 4. TMAO and cardiovascular aging. Certain gut bacteria metabolize compounds found in red meat and eggs to produce TMAO, trimethylamine N-oxide. Elevated TMAO levels are strongly associated with accelerated atherosclerosis and cardiovascular disease. Interestingly, plant-based diets and Mediterranean diets produce microbiome profiles that generate far less TMAO, which may partly explain the cardiovascular longevity benefits seen in populations following these dietary patterns. The Centenarian microbiome studies are some of the most compelling data in longevity research. Let me highlight the key findings. A 2021 study published in Nature Metabolism analyzed the gut microbiomes of 160 Japanese centenarians and found unusually high levels of bacteria producing secondary bile acids with antimicrobial properties, suggesting the microbiomes of long-lived individuals actively suppressed harmful pathogens. The centenarians also had higher diversity of allostipes and other fiber-fermenting species. Research on Sardinian centenarians from one of the world's 5 blue zones found elevated levels of Akkermansia muciniphila, a species increasingly associated with gut barrier integrity, metabolic health, and reduced inflammation. Multiple studies now associate higher Akkermansia abundance with lower rates of obesity, type 2 diabetes, and cardiovascular disease. Across studies, two patterns emerge consistently in long-lived populations: higher microbial diversity and a higher ratio of beneficial to potentially harmful species. Both of these decline with conventional aging, but the centenarians buck this trend. Crucially, this is not simply genetic. When researchers have transplanted microbiomes from aged mice into young germ-free mice, the young mice showed accelerated biological aging markers. When the reverse was done, young microbiome transplanted into aged mice, the aged mice showed cognitive improvements and reduced systemic inflammation. This is a powerful demonstration of causality, not just correlation. I want to spend a moment on Akkermansia muciniphila because the research on this species has accelerated significantly in the last 3 years. Akkermansia lives in the mucus layer of your gut, where it feeds on mucins and in turn stimulates the gut to produce more mucus. This maintains the physical integrity of the gut barrier. Higher Akkermansia abundance is consistently associated with a healthier gut lining, reduced intestinal permeability, and lower systemic inflammation. A landmark 2021 clinical trial by Patrice Caney's group in Belgium, published in Nature Medicine, found that supplementation with pasteurized Akkermansia in overweight adults improved metabolic markers, reduced liver inflammation, and improved gut barrier function. This was a randomized, double-blind, placebo-controlled trial, the gold standard in clinical research. Pasteurized Akkermansia, meaning heat-treated, not live, is now available as a supplement and has a stronger evidence base than many probiotics currently on the market. It is also more stable because live Akkermansia is extremely difficult to keep viable outside of the gut environment. I have reviewed the available Akkermansia supplements against the clinical specifications used in the Belgium trial. The link is in the description below. Let me give you the evidence-based interventions, ranked by strength of evidence. Number 1. Dietary fiber diversity. The single most consistent finding across microbiome research is that diverse plant fiber drives microbial diversity. Aim for 30 or more different plant foods per week. Not 30 servings, 30 different types. This includes vegetables, fruits, legumes, nuts, seeds, herbs, and whole grains. A 2018 study from the American Gut Project found that people eating 30 or more plant types per week had significantly more diverse microbiomes than those eating 10 or fewer. Number 2, fermented foods. A 2021 Stanford study published in Cell found that a high fermented food diet, yogurt, kefir, kimchi, sauerkraut, kombucha, significantly increased microbial diversity and reduced 19 inflammatory proteins in just 10 weeks. This was a randomized controlled trial. The effect was larger than the high fiber diet group. Fermented foods work. Number 3, targeted probiotics. The evidence for probiotics is strain-specific. Generic products with undisclosed strains have weak evidence. Look for products with clinical evidence for specific strains. Lactobacillus rhamnosus GG for gut barrier function, Bifidobacterium longum for neuroinflammation, Akkermansia muciniphila for metabolic health and gut integrity. Number 4, prebiotics. Prebiotics are the food for beneficial bacteria. The best evidenced prebiotics are inulin, FOS, fructooligosaccharides, and resistant starch. These selectively feed Bifidobacterium and Akkermansia species. Foods high in these compounds include chicory root, Jerusalem artichoke, raw garlic, leeks, slightly underripe bananas, and cooked then cooled rice or potatoes. Number 5, avoid unnecessary antibiotics and limit ultra-processed foods. Antibiotics cause significant and sometimes lasting shifts in the microbiome. Ultra-processed foods suppress microbial diversity and feed pathogenic species. These are not lifestyle preferences. They are microbiome interventions with documented downstream health consequences. The gut longevity connection is one of the most exciting frontiers in health science right now, and it is practical. You can begin shifting your microbiome today through dietary choices before any supplementation. On Monday, we are going back to cellular biology. Specifically, we are looking at chronic inflammation, what it is at the molecular level, why it accelerates aging, and 7 natural compounds with strong research behind them for reducing it systemically. That is a video worth setting a reminder for. Subscribe if you want the science delivered properly, twice a week. All references are linked below. See you Monday.